Good morning, dear students. Today we are going to a new chapter, Nature of Genetic Material. First, search for the genetic material. DNA is first isolated from where? It is isolated from human pus cells. Who isolate the DNA for the first time? By Frederick Mitchell in 1969. And he gave it the name nuclei. Why he named like that nuclei? He think that it is something that comes out from the nucleus, and that time he doesn't know what is that. So he simply give that name nuclei. And later it was found that it is an acid, and along with that phosphorus also present, and renamed as nucleic acid. Okay. Then what would be the nature of a genetic material? Misha first believed that is something that is passing from one cell to another in the form of a chemical code stored in proteins. First he think like that. And generally there are four general requirements are there to become a genetic material. What are those general requirements? Well, we can see. First requirement is that whatever it be, it should have the capability for self-replication. It should have the capability for self-replication. Second requirement is that it should possess a stable structure. Whatever it be, that genetic material should possess a stable structure. The third one is that it should have the potentiality to carry all the necessary biological informations. That is the third requirement. What is the fourth? Along with all the three requirements, it should be able to transmit the information from one cell to other cell. These are the four general requirements. By keeping all these requirements in mind, scientists are engaged with experiments to find out which is the genetic material, which component stands as the genetic material. The primary candidate they considered is proteins. Why they consider proteins as the primary candidate to become a genetic material? Because proteins are formed of amino acids, and 20 different amino acids are available to form different combinations of protein. So they consider protein as the primary candidate. And that time they didn't even consider DNA as a second candidate itself. Why? Because they know that DNA is formed of only four nucleotides. Which are the four nucleotides? Adenine, thymine, cytosine, guanine nucleotides. These are the four nucleotides. With only these mere four nucleotides, how can form such a complicated compound that can transform genetic material from one cell to another? So they didn't consider DNA as a candidate to become genetic material. But later, scientists have identified DNA as the genetic material. Who found that for the first time? It is Frederick Griffith. In 1928, he is a British medical officer. He had done an experiment in Streptococcus bacterium. Next, we are going to see what is his work. Evidences for DNA as the genetic material. That is Griffith's experiment on where pneumococcus bacteria, and that itself is a type of transformation. For your examination, there is more chance to have this type of questions. I explain the Griffith's experiment to prove DNA as the genetic material. So don't skip this part. So first we need to know what is pneumococcus. It's a bacteria which can cause pneumonia in rat. And those rat which were infected by this bacteria will surely die because of the pneumonia. And this pneumococcus exists in two forms. Two strains are there. Which are the strain? Smooth strain and rough strain. What is the difference between the smooth strain and the rough strain? And also the smooth strain is otherwise called S strain or S3 strain. Similar to that, rough strain is also known as R strain or R2 strain. The smooth strain is the virulent. Virulent means the disease causing one. So that is pathogenic. And what is the peculiarity of the smooth strain? Peculiarity is that it has an outer polysaccharide coat. Bacteria, smooth strain bacteria has an outer polysaccharide coat which protect this bacteria from being killed by the host. So it can survive in the body of the host and can cause pneumonia. Okay, that is the peculiarity of smooth strain. When coming to this rough strain, it lacks this polysaccharide coat. So, it will be killed by the host cell. So, it cannot be virulent. It is avirulent and not pathogenic. That's about rough strain. So, next we can go and see what is the experiment done by Griffith. Griffith's experiment. He took one rat and injected it with a smooth strain of pneumococcus. And what will be the result? That rat died because of pneumonia. And later he took another rat and injected this rat with a rough strain of pneumococcus. But this rat survived. Okay, that means this rough strain cannot cause pneumonia in this rat. He again repeat the experiment by taking another rat and inject this rat with a heat-killed smooth strain pneumococcus. Heat-killed means it lost the property of virulence. It can't cause pneumonia in the rat, so the rat survived. Okay, then what he did was he inject rough strain along with heat-killed strain to the body of a rat. Then what he found that rat found dead again, and from the body of this dead rat he could find the samples of smooth strain pneumococcus bacteria. Okay, then what is the conclusion of this experiment? He concluded that from that heat-killed S strain, some properties transferred to the rough strain and transformed this rough strain to a smooth strain. And what is that property? And that property he called it as transforming principle. A transforming principle. And he doesn't know what is that thing, that convert that rough strain to a smooth strain. But he know that whatever it is, it will contain the instruction to synthesize the polysaccharide coat in the rough strain. So that is because that rough strain turned to a smooth strain. But he cannot understand that transforming principle is DNA that time. Okay, then later O.T. Avery, C.M. MacLeod and M.C. Carty, they found that transforming principle was nothing other than deoxyribonucleic acid, that is DNA. Okay, how they find that? They took some heat strain pneumococcus bacteria, and from those bacteria they isolated its DNA and add this DNA to the rough strain pneumococcus bacteria, and because of that rough strain get transformed to a smooth strain. And then they could identify that the DNA is the transforming principle responsible for converting the rough strain to a smooth strain. And that phenomena of acquisition of new genetic characters by a cell, it is called transformation. That rough strain pneumococcus have changed to smooth strain because of the acquisition of new genetic material. Okay, and that process is called transformation. Then what are the contributions of Avery, MacLeod and MC Carty to substantiate that DNA is the genetic material? That we are going to see next. This is a bacterial cell and they treat this bacterial cell with some organic solvents and denature almost all proteins. And still that bacteria is found to carry that transforming or transformation property. That means proteins didn't have any role in transforming the genetic material from one bacterial cell to next. Then they treat the bacterial cell with enzymes like trypsin and chymotrypsin, and by that way they denature some other proteins also, and still that bacterial cell is found to show that transforming property, and further they avoided or discarded the role of protein as genetic material. Then they treat the bacteria with some RNAs. RNAs are the enzymes that denature RNA particle, whatever it present in the bacteria. And after denaturing and destroying all those RNA particles, still then this bacteria is showing the transforming property. So they again discarded the role of RNA as the genetic material. Then they use a DNase. DNase is the enzyme that can denature DNA. And after using or applying a DNase, they found that the bacterial cell lose the transforming property. That means the DNA is the genetic material. Okay, and still here they didn't stop. Instead, they further do some more experiment to affirm that DNA itself the genetic material. What are the further work done by them that we are going to see? They isolated that substance that the substance that they are expected to be as the genetic material. That substance they isolated, and they do ultracentrifugation for that substance and found some very high molecular weight substance, and that substance is nature also somewhat similar to the DNA, and that inference it further strengthened the belief that DNA is the genetic material. Then what they did was they take that substance and give that substance some electric field, and found that that substance show high motility. That also is another important behavior of DNA. So again it add weight for the belief that DNA is the genetic material. Then they perform UV spectrophotometry for that substance and found that their range of absorbance for the wavelength is same as that of the DNA. That further strengthened the belief that the substance is DNA. And then they examined the nitrogen to phosphorus ratio of that unknown substance, and they found that that ratio is also same as that of the DNA. And in all these way, they have further proved that the substance that is conveying genetic material or transforming genetic material from one bacterial cell to next is DNA itself. Okay. Then we are moving to another set of experiment performed by Conrad and Stearns. They did their experiment with the TMV virus, tobacco mosaic virus. What the intention and what was the outcome of their experiment? The outcome was to prove, they proved that through that experiment, they proved that RNA also sometimes be the genetic material. First, we need to know about tobacco mosaic virus or TMV virus. They didn't contain any DNA. Instead, it is formed of simply protein and RNA. So, that virus is called an RNA virus. So, next we can go and see what experiment they perform using this TMV. For performing that experiment, they took a TMV type virus and HRV type virus. and then they denature these two viruses and separates its RNA component and protein code component, means protein component. Here also is RNA component and protein code component are separated. Okay. After degradation or that separation, then they mix these two varieties together. And because of that mixing up, a hybrid form of virus is formed, and it is having the RNA of TMV virus and protein coat of HRV type virus. Such a hybrid is formed because of that mixing up. And this hybrid virus is permitted to infect a leaf, and that leaf develop lesions. And in the lesions, then they later examine to find out which type of virus present in those lesions and found this type of virus, that is a TMV progeny is present in these lesions instead of this hybrid variety. Why? Because here in this hybrid, the RNA that present here is that of the TMV virus. Okay. And the protein code here is from the HRV. Okay. But look in this progeny. The progeny, instead of the protein code present in HRV, it developed the protein code of a TMV. Why? Because in this hybrid that is used to infect the leaf have the RNA from TMV. So, as that RNA is present, it synthesizes the protein code according to the information present in that RNA. That means the RNA of this TMV. Clear? By doing that experiment, they proved that it is not protein, but the RNA itself is acting the genetic material. Okay, that is their experiment. Next, we are going to see Hershey and Chase experiment in bacteriophages. They performed that experiment in T2 bacteriophage. For what purpose they did that experiment? To further substantiate that DNA itself is the genetic material. Then we need to understand what is bacteriophage. Its image is shown here. It is a virus attacking bacteria. And what are its parts? Its parts include a head and a tail. And regarding the head, it contains a central core of DNA, and it is covered by a proteinaceous coat called capsule. And the tail is composed entirely of protein. That is its parts. Then regarding in the way how they infect a bacterial cell, that I will explain to you. Suppose this is a bacteriophage, and this is going to infect a bacteria. And for that, first it attaches the body of bacteria using its tail. After attaching the cell wall of the bacteria, the next step is that it injected its genetic material. What is that? Its DNA into the bacterial cell. Okay. That it injects its DNA into the infected bacterial cell. Along with that, the bacteria itself possesses its own genetic material. Circular DNA also will be here. And as this DNA from the phage enters the bacteria, it undergoes multiplication inside the bacterial cell. And using the machinery present in the bacteria, later it formed the protein coat of this phage, protein coat for this phages. And after forming all this, it breaks, it lyses this bacteria, and then it comes out. Yeah, the next progeny phages ready to attack another bacteria. This is its mode of infection. I think that you understand what is the mode of infection of a phage, bacteriophage to a bacterial cell. Okay, then next point is about the objective of the experiment. Um, whether the main intention is to prove that whether that phage DNA or the phage protein, or whether they both are necessary for the phage reproduction. That need to be proved. That is the objective. And the target point, based on what principle they did that experiment, is they mainly focused on the chemical difference between DNA and protein. The main difference they consider here is that the DNA only possesses phosphorus and the protein only possesses the sulfur. And they take advantage of this property, this difference between the DNA and protein. Then we can directly go to the experiment. For this, the scientists prepared two types of T2 bacteriophages. And one is carrying radioactivity in the protein coat, especially in its sulfur. Sulfur of its protein coat is radioactively labeled. And the next one, the DNA of the next phage, it is radioactively labeled in its phosphorus. And they take these two T2 bacteriophages to infect bacterial cell and allow them to infect the bacterial cells. Okay, they infected this bacterial cell. After they infect, then the scientists carried out a centrifugation for these two types of cells. What's the purpose of doing that centrifugation there? Is to separate this attached bacteriophages from the cell wall of the infected bacterial cell. For that, they perform that centrifugation. And we know that because of centrifugation, in the centrifugation tube, I am drawing two tubes to explain both these cases. And in this case, we know that because of centrifugation, those substances having high density will settle in the bottom, and those having low density will settle in the top. So here also, since the bacterial cell having high density, it settles in the bottom. Okay, and the phage cells, since it has low density, it is settle in the top. And in this case, as they examine for the radioactivity, they cannot find any radioactivity in the bacterial cell that settled in the bottom. They can find that radioactivity only in the phage in this case. Then coming to the next one, here also depending upon the density, bacterial cell settles to the bottom. Bacterial cell settles to the bottom, and phage cell here also we can see on in the top. Okay, and here they examine to find the location of radioactivity, and in this case they find the radioactivity is located inside the bacterial cell, and here there is no radioactivity in the phages. Okay, clear? And what conclusion they got out of this result? The conclusion is that the thing that is transferred to the infected bacteria to form the next progeny is DNA, and it is not the protein. Okay. Here also the DNA has transformed to this bacterial cell, but in this case, as its DNA is not radioactively labeled, that is why the scientists could not find any radioactivity in these bacterial cells. Okay, but in this case, as the DNA of this phage is radioactively labeled and it get transformed to the infected bacterial cell, they can locate radioactivity in the infected bacterial cell. And where the new progeny is going to be formed. So by that way they prove that it is DNA and not protein is the genetic material. Okay, that's about the next picture. No radioactivity enters inside the bacterial cell. Here radioactivity we can locate, because here in this phase that radioactivity is given to the DNA. That is why in the settled bacteria we can locate radioactivity inside. Okay, this is the experiment done by Hershey and Chase to further substantiate that DNA itself is the genetic material. Okay, with this we can finish today's class and section. Thank you.
I'm an icon, man. And my girl about a 10. I should fuck her in a Benz.